Hi everyone. I have the great pleasure of introducing Dr. Kathleen Gavin as today's nutrition and food science speaker. Dr. Gavin received her bachelor in health and exercise science from Syracuse University and her master's in integrative physiology from the University of Colorado. She received her Ph.D. in bioenergetics and exercise science from East Carolina University. Her dissertation studies investigated the influence of estrogen on metabolic function and body fat distribution in premenopausal women. She pursued her postdoctoral research at the Division of Geriatric Medicine in the University of Colorado Anschutz Medical Campus, where she is now an assistant professor. Her lab currently works on translational studies of fat cell lineage in humans, and her overarching research interest is how the origin of fat cells may play an important role in sex-specific body fat distribution and obesity-related cardiometabolic disease risk. Her most recent work and topic of her talk today is on the development of distinct adipocyte populations. Please join me in welcoming Dr. Gavin.
Thanks so much for the invitation to talk today. Dr. Giles invited me, and it's great to have the opportunity to come check out your huge campus and be able to talk to you about the research that I started doing as part of my postdoctoral fellowship in the labs of Dr. Wendy Court and Dr. Dwight Clem, and have now been able to continue as part of my faculty appointment at the University of Colorado. So, today I'm going to be talking to you about the development of fat cells and the development of distinct and specific types of fat cells that we think are playing an important role in disease risk. So quick run through of the outline. First I'm going to talk about adipose tissue background, then about the different lineages and development lineages that we know happen in adult humans. So this talk will mostly be focused on what we know clinically, although I will present a variety of basic studies. So what I'm going to focus my talk on is the existence of bone marrow progenitor-derived adipocytes. So although the name of them starts with the words bone marrow, these are actually adipocytes that are found in the adipose tissue. So it's actually the origin of where their progenitors come from, where the word bone marrow comes from, and I'll get into the details of that. But want to lay that out, we're talking about the major white adipose tissue depots in mice and in humans. Then I'll go into the factors influencing the production of these cells, talk about some of my ongoing research, and then finish with some conclusions and some of my future directions. So I think adipose tissue is a fascinating organ, and a lot of that has to do with its extraordinary capacity to shrink and expand that's unmatched by any of other organs in the body. So most of us talk about adipose tissue in the context of obesity, and we're all very familiar with the idea that carrying around excess adipose tissue results in a variety of chronic diseases and disease risk, metabolic syndrome, cardiovascular disease, a variety of types of cancer. But what many people don't often think about is the opposite end of the spectrum, and lipodystrophy syndromes in which people are unable to store adipose tissue in the places in the body where it's usually stored. And this leads to storing excess nutrients and lipids in places such as muscle and liver. And this also results in a wide variety of disease risks. So hypertriglyceridemia, insulin resistance, liver problems. So really, there are extreme consequences on both ends of the spectrum. So the point I want to make here is just being able to store adipose is really important to maintaining physiological homeostasis. So you need some, and you need to store it in the right places in order to kind of stay healthy. So in my research, I'm really interested in body fat distribution. And I'm in the Division of Geriatric Medicine, and I'm really interested in studying consequences of aging and how that changes where you store your body fat. So one of the primary things I'm interested in, as was mentioned in my introduction, is kind of how estrogen and sex hormones impact where you store your body fat. So one of the main things I'm interested in looking at is how women in the premenopausal state tend to store their weight in their hips and thighs. So they're more of a pear-shaped body fat distribution. This peripheral or subcutaneous body fat distribution is associated with lower risk for chronic diseases such as cardiovascular disease and diabetes. As women lose their ovarian hormones and go through the menopausal transition, you can see they become more apple-shaped. And this is more of a body fat distribution that's observed in males throughout life. So even if women don't gain weight through the menopausal transition, they tend to shift where they store their fat from their hips and thighs into their belly, and predominantly in the visceral depot. So this is an area where we know it's not so healthy to store your fat. So I'm really interested in why does this happen, and how do sex hormones regulate where people store their fat in men and women? So this is a good example in women. Men don't tend to see this really dramatic shift. They tend to carry it more in their belly throughout life, but I still think sex hormones and the amount of testosterone compared to estrogen may have some influence in where men store their fat. Adipose tissue is also really interesting because it's not just an inert energy storage site, but it's also an endocrine organ. So we now know it secretes a variety of factors called adipokines that traffic out to have influence on many different systems throughout the body. So some things are logical: appetite and energy expenditure, or insulin sensitivity and glucose homeostasis. But the adipose tissue also secretes factors that influence the generation of new blood vessels, as well as inflammation and immunity. So it's a very complex organ, and we need a lot more research to understand exactly how the components of the adipose tissue are impacting overall health. One other thing many people don't really understand is that adipose tissue is not just made up of fat cells. So although by volume, adipocytes or fat cells are the primary component of adipose tissue, there's also a variety of other types of cells that I have here at the bottom that are dispersed throughout the adipose tissue. And so these other non-adipocyte cells make up what is called the stromal vascular fraction of the adipose tissue. So depending on the depot or the disease state, there may be more or less amounts of these non-adipocyte types of cells in the adipose tissue, contributing to the overall phenotype of that depot. You also have local blood vessels and blood flow that play a role in the phenotype of each individual depot. And you also have neural innervation. So there's a variety of things going on in the adipose tissue that combine to make an overall microenvironment and have each individual adipose tissue depot act a little bit differently. What I'm really interested in is the concept that within an adipose depot, that there's actually, I don't think the colors show up really well on here, but there are different types of adipocytes that are located within a singular adipose tissue depot. So not only do you have differences whether it's fat that's in your belly or in your hips and thighs, but I think within one of those depots, there's actually different types of fat cells that might have different characteristics and phenotypes that are releasing different types of adipokines or have different insulin or insulin sensitivity that may be a really important factor in the phenotype of that depot. So let's talk for a minute about the different types of adipocytes that we know humans have. So I'm going to start with the one in the middle on the bottom here, the conventional white adipocyte. And you'll hear me refer to a conventional adipocyte throughout the talk. This is what I'm talking about. This is the fat cell most people think of when they think of an adipocyte. So it's a single unilocular lipid droplet. Primary function is to store nutrients and mobilize them as needed, also secreting adipokines. So this is kind of our primary subcutaneous or visceral type of adipocyte. On the other end of the spectrum, we have what are called brown adipocytes. And so for many years, you guys are probably familiar with this, it was only thought that rodents actually have brown adipocytes. These adipocytes are thermogenic, so they generate heat, and they get their name as a brown adipocyte because they have a high mitochondrial content. So when you look at them under a microscope or on a slide, you see that they actually look more brown than our conventional white adipocyte. So these cells have many lipid droplets, higher mitochondrial content, and a high expression of uncoupling protein 1, or UCP1. So these cells, the uncoupling protein uncouples the electron transport chain, and instead of generating coupled ATP at the end of transporting electrons down the ETC, you get the generation of heat. So this is a novel area that people are starting to look at for trying to regulate human body weight. So I'm not going to talk about brown adipocytes, although they're very interesting. And then the in-between cell type is this this beige adipocyte. So this doesn't have quite as much mitochondria as a brown adipocyte, has a few more lipid droplets than a unilocular white adipocyte, but it has more of a thermogenic capacity. So this type of adipocyte is considered more similar to a white adipocyte, and it seems, or it is believed to be developed from a similar mesenchymal lineage stem cell that a white adipocyte is. So dogma in adipose tissue biology for many years has been that all new adipocytes arise from mesenchymal lineage stem cells present in the adipose tissue. They self-renew as a new fat cell is needed, commit to the adipocyte lineage, and differentiate into a new fat cell. So we now know that both white and beige adipocytes can come from this mesenchymal lineage. We also know that the human body is always turning over its fat cells. So fat cells live for about 10 years, and then a new one needs to take its place, whether you're gaining or losing weight. So this is a constant phenomenon. You need to have this pool of progenitor or stem cells in your adipose tissue ready to make new fat cells. So as more studies have been done on the brown adipocyte, people used to believe it came from the exact same mesenchymal lineage cell as a white adipocyte because they're both fat cells. What we've recently found out is that they still come from the mesenchymal lineage of stem cells, but they actually come from a slightly different lineage within the mesenchymal lineage. And it's actually a lineage that's more similar to the type of stem cell that would produce a skeletal muscle cell. So a skeletal muscle cell has much higher mitochondrial content than an adipose tissue cell would have. So we know that this makes sense. These types of cells are very similar. So now we know just within the mesenchymal lineage that it's not quite as simple as we always thought. There's one type of stem cell that creates all new fat cells. So in recent years, our ability to trace how cells develop has really advanced, and there's a number of groups that are working on this. And one group has recently discovered the idea that maybe endothelial lineage cells actually contribute to the production of new fat cells. So these are cells that line the blood vessels of the small capillaries that are in the adipose tissue. And so they are not a mesenchymal lineage cell, but they're actually a different lineage cell that some labs have discovered do actually generate potentially brown, white, and beige adipocytes. So I'm not going to talk about this today, but I want you guys to get a sense for this is not as simple a concept as people once thought it was. What I'm going to talk about is how fat cells might develop from the hematopoietic lineage. So these are stem cells that are produced in the bone marrow, and their primary purpose is to generate different types of blood and immune cells. So they're in the bone marrow, tend to be a CD45 is a kind of an overall marker we tend to use for cells that come from the hematopoietic lineage, at least in humans. And we know these cells are in the bone marrow, and they differentiate into a precursor cell or a progenitor cell many times of the myeloid lineage. So these tend to be cells that would ultimately differentiate into an end-stage immune or inflammatory cell. So these myeloid lineage cells, as part of their normal function, traffic through the blood. And we also know that these myeloid lineage cells actually end up in the adipose tissue. We've known this for a long time. So many of you who study obesity are familiar with the concept that as you accumulate more adipose tissue, you may have a more inflammatory phenotype of that adipose tissue. And it's believed that more immune cells are accumulating in your fat tissue as you gain more adipose. So we know that these myeloid lineage cells are present in the adipose tissue. What was never researched or believed previously was that these cells can actually transdifferentiate in the adipose tissue into a mesenchymal-like precursor that can then differentiate into a mature fat cell. And we think it's a fat cell that has a unique phenotype and gene expression fingerprint that may be having an important impact on the overall phenotype of the adipose tissue depot. So the next thing I'm going to present to you is kind of some of the original research that was done by my basic science mentor, Dwight Clem. So he was the one with this original hypothesis. So he's an adipocyte biologist, and he was really wondering, could non-resident or bone marrow progenitor cells be recruited to the adipose tissue and actually contribute to adipogenesis or making new fat cells? We know the cells are there. Is it possible that they could play this role? So he's a basic scientist, and he uses a lot of really neat lineage tracing models. And so I'm going to just, at a very surface level, describe some of these for you. So in order to determine where these fat cells come from and to do lineage tracing studies, if he thinks they're coming from the bone marrow, he needs to find a way to trace them from where they start their life to where they end up as a mature differentiated cell. And one way to do this is to perform a bone marrow transplantation. So you take an animal that is a transgenic animal that has ubiquitously expressing green fluorescent protein, yellow fluorescent protein, and you can take the bone marrow out of that animal, transplant it into an irradiated mouse that then the bone marrow will engraft into. Then you can let that mouse grow and live for however long, and then a certain number of weeks or months later, you can take that animal and measure if there's any cells in your tissue of interest that express that tag that would have only been on the transplanted bone marrow cells. And so this is what Dwight did for his initial experiments that he published about 10 years ago now. So he took some mice, transplanted them with a green fluorescent protein, waited 8 to 16 weeks, took out some fat pads, and you can see here this is a tissue digest of floating adipocytes. And you can see some of them have these really bright green membranes. And this would be evidence that some of the fat cells in this tissue digest have the green fluorescent protein that was restricted only to the cells in the bone marrow. So this was the first evidence that after about 8 to 16 weeks, there's bone marrow progenitor derived, or BMP derived, adipocytes that are comprising about 5 to 25% of the adipocyte population in these mice. Another technique that Dwight uses is a luciferase model. So this is a transplantation where you're able to take bone marrow that has a luciferase, which is a light emitting enzyme, that is driven by an adiponectin promoter. So this is a gene that is highly specific for adipocytes. So you can take these cells and they will only express luciferase when and if they express adiponectin. And this allows him, instead of having to take out the fat depots of the animal and measure GFP by either a DNA or a microscopy or flow cytometry measurement, you can image the animals in vivo over time and be able to see the luminescence of the development of these new fat cells. So each of these different pixels here, and as you see the brighter yellow and red colors, indicate the production of new fat cells that are emitting this luciferase. So this is a way for us to study these over time, and you can see this goes from 2 to 18 weeks post-transplant, how the accumulation and production of these cells increases. So this is really handy for us to be able to do these studies on animals and not have to collect their tissues till the very end. So what are some of the first things that he found? Well, we were able to have animals using this luciferase model. We took the different depots out of the animal, so the major fat depots of the mouse, including gonadal, subcutaneous, perirenal, mesenteric, and the fat around the heart, so peri or epicardial, and then as well as some other organs of interest, just to see if this luciferase is really showing up in the depots that we expect it to. And sure enough, we found, yep. So yeah, there's no brown fat on here. We have looked a little bit at interscapular fat. We don't have that quantified on any of our current things. We do think that they're not contributing a lot to the brown fat depots, but yeah, we don't have those on here. So you can see that the two depots that have the highest accumulation are the epi or pericardial as well as the gonadal depot. And these were of interest because these are regions where we know it's not so healthy to store your fat. So these are some of the more intra-abdominal or visceral cavities that you see in the mouse. So we don't see a super high expression in the mesenteric, although we've had a hard time really getting a good sized fat depot of that out in the mouse. So we think that's why it's so low there. But we repetitively, and you'll see throughout my talk, see a really high expression in the gonadal tissue. And I don't have much more to talk today about the peri and epicardial, but we're really interested in that and understanding how that may influence cardiovascular disease risk. So other important point here is that we really don't see any detection in these other tissues of interest, showing that our model is really specific to the cells that have the adiponectin gene expressed. So Dwight has done gene arrays on bone marrow derived adipocytes in comparison to conventional white adipocytes. And what he's found is that there's a variety of inflammatory cytokines that are anywhere from 50 to about 2.5-fold more highly expressed, as indicated by the red color here, in the bone marrow derived adipocytes as compared to the conventional. And on the other hand, he's found that leptin, which was the original adipokine, a factor we know that is secreted from adipocytes in high amounts, is really low in the bone marrow derived adipocytes. So this was a very surprising finding as far as low leptin and high inflammatory cytokines, although it goes along with where we're finding these cells, in the depots where you would expect this to be the characteristics of these cells. He also found that mitochondrial and peroxisomal enzymes were also more lowly expressed in the bone marrow derived compared to conventional white adipocytes. So all of this is adding up to him believing that these cells are really having a negative metabolic or a not so great metabolic phenotype that may be influencing disease risk. Through the gene array analysis, he's also conducted principal component analysis to identify the fact that these BMP-derived adipocytes tend to cluster with both white and brown adipocytes as compared to a peripheral blood myeloid cell or a stromal vascular myeloid cell. So these would be the cells we believe are the progenitors for these fat cells. But when they undergo differentiation, they are becoming a bona fide adipocyte. However, when we are able to just do the component analysis just with the different lineages of adipocytes, we see the bone marrow-derived cells really cluster over here by themselves. So they are different and a distinct lineage than either the white or the brown adipocytes. So really piqued our interest just so, although we kind of lump them a little bit more in with the white adipocytes, they do really have their own kind of phenotype. Well, according to their gene expression, we have a ways to go on the phenotype. So this is the developmental model that Dwight published back in 2010. So this is a schematic of kind of everything I've been talking through so far. So these progenitors are produced in the bone marrow. So this is where they start their life. At that point, they're a hematopoietic progenitor, and in humans, they'll express CD45. They differentiate down the myeloid lineage, and this is where they start to express their different myeloid lineage surface markers. And that's what these little blue and red dots are on the surface here. So these cells leave the bone marrow, traffic through the blood, and end up in the adipose tissue. And at this point, they differentiate into a myeloid-derived adipocyte progenitor. So these are cells that now kind of look like a mesenchymal stem cell. So they lose their markers when they go through this transdifferentiation, saying they came from the bone marrow, which makes our studies in humans pretty challenging, but I'll get into what we're doing about that in a minute. But in the animals, we still have these fluorescently tagged or luminescent cells that we're able to measure and know that they came from the bone marrow. So this is kind of the overall schematic of what we believe is happening with the development of these cells. So some signal is telling these cells to end up in the adipose tissue, and then the right signals are there in the microenvironment to tell these cells that they can go down a differentiation pathway that they usually wouldn't ever go down. So what about the clinical relevance of this? So, I had two mentors as part of my postdoc, and they were both very instrumental in moving this research forward. And it was my mentor, Dr. Wendy Court. She's a clinical researcher. She attended one of Dr. Clem's talks, and he was talking about these cells, and I'm going to get to some of the sex differences that he's found in his animals. But she, similar to me, is really interested in how body fat distribution changes in women with age. And some of his findings with these new cells piqued her interest. And so they decided to write a grant together, and I was able to come in as a postdoc to take over this project, trying to translate the measurement of these findings that he had in his animals into humans. And how do we do that? Because we don't have a surface marker saying, hey, this is a cell that came from the bone marrow, and we also can't do fluorescently tagged bone marrow transplants in humans. So what we were able to do was come up with a collaboration with our bone marrow transplantation clinic at the University of Colorado Hospital. So Dr. Jonathan Gutman directs this clinic, and he has a variety of patients who have had a bone marrow transplantation for a number of different clinical indications, a variety of blood cancers. So we were able to recruit some of his patients who had had a previous bone marrow transplantation, but were now thriving and doing well, and were willing to come in and have a subcutaneous adipose tissue biopsy taken from them. So the method that we used was a chimerism analysis. So how bone marrow transplantation engraftment is measured is by measuring different specific parts of single nucleotide polymorphisms of the DNA of the donor of bone marrow in comparison to someone who is receiving the bone marrow transplantation. And so clinically, when somebody goes in, you know the DNA signatures of the two different people, and then once they receive the bone marrow transplantation, a certain number of time later, they will have a blood or a bone marrow aspirate taken. They measure the DNA at SNPs, and they determine how many of the cells either in the blood and the bone marrow now have the DNA signature of the donor. And what you want to see is 95 to 100% of these cells now being from the donor. So it's a now thriving new bone marrow environment that can generate new healthy blood cells. So all of the patients we enrolled were at least 95% engraft. Most of them, I think almost all of them, were 100% engrafted. And we were able to obtain, again, a subcutaneous abdominal adipose tissue biopsy from right next to the belly button. And everyone was at least 1 year after their bone marrow transplantation. So we took out the fat sample, and then as you saw previously, the fat has a lot of different cell types in it. And a lot of those are cells we would expect to come from the bone marrow. So we needed to get a really clean isolation of just fat cells. So we used a variety of techniques to isolate out the fat cells and do some flow cytometry sorting of intact adipocytes in order to perform this chimerism analysis by the clinical lab that performs the same DNA measurement for the clinical engraftment study. So we were able to use the clinical DNA measurements from the patient's transplant and compare them to the DNA that was extracted from their fat cell. And if we found donor DNA, that would be our evidence that yes, indeed, some of these fat cells came from the bone marrow. So we brought in about 8 patients, and the way I'm going to present the data here is on the y-axis we have donor chimerism. So this is the percent of cells that we found had evidence of donor DNA. So the higher the number, the more cells came from the donor, okay? And then we measured these people at a variety of times post when they had their transplant. So some people were 1 year past, one person was about 7 years past. So we have a variety of times. And we also have a few people that we brought in for more than one biopsy. So what did we find? Well, we found that in some people it was 0 to less than 5% of adipocytes had evidence of donor DNA, but in one subject there was about 35% of their cells had evidence of donor DNA. And there was a pretty good relationship with time since transplant, so these bone marrow derived cells are continually making new fat cells after they have their transplantation. These colored dots also indicate people that we brought in for repeat biopsies. And you can see that there's a similar trajectory in all of these people. If we brought them in 6 months to a year later, we see an increase in the percentage of cells that had donor DNA. So this was really exciting and really the first evidence that, we thought would be out there about these cells existing in humans. But little did we know, at the same time, there's always another group that's working on exactly what you're doing. So Dwight's animal studies had been published for quite a few years. And there was another group out of the Karolinska Institute. If you do adipose tissue research, you've heard of Peter Arner, and he's the one who discovered the turnover rate of adipocytes in humans. Very well-respected group. And we didn't know it, but they were conducting basically the identical study to this. We went to a conference and presented in the same session, like pretty much the same data. But they have a much larger sample size, so their healthcare system there allows them access to very many more patients. So they were able to bring in 65 patients, anywhere from 3 to 35, almost 35 years after their transplant, and they once again saw a similar significant correlation that time since transplant you do see an increased production of bone marrow derived adipocytes. So this was great, and a lot of this work in the adipocyte world has been a bit controversial because people don't want to believe that the way we've always thought fat cells developed might not be the way it is. And so to have this work replicated and have it shown in a clinical population has been really key in this taking a foothold and people understanding the potential clinical importance and why we need to keep studying this in a human population. So these studies were both published last year. So what have we talked about so far? Well, we know that marrow-derived progenitors appear to contribute to adipogenesis in mice and humans. We also know that they typically constitute less than 20% of the total fat cells, at least in humans, but we hypothesize that they're still physiologically relevant. So we know that there's small number of fat cells that are different types of immune cells that can infiltrate the fat tissue at lower amounts and still have a physiological impact. So we do think that this percentage is of physiological importance. We also have observed that they have a unique gene expression fingerprint and production pattern. So they appear to be more in central depots, but present in the major white fat depots. Notable, we are taking subcutaneous adipose tissue biopsies from these bone marrow transplant patients. So we would hypothesize if we could get a visceral or more deep adipose tissue sample that maybe the percentage that we're observing of cells that are coming from the bone marrow might be a little bit higher in the visceral region. So what are some of the things we've come across so far in our research that might actually influence the production of bone marrow derived adipocytes? Well, I'm going to talk about 2 today. One is obesity status and the other is ovarian hormone status. So the obesity findings are actually from the clinical paper by Peter Arner's group that was published last year. Since they had a much larger sample size, they were able to break down into obesity status. And then I'm going to present some data from our group about the ovarian hormone status, and this is kind of the direction my research is moving in. So what did Peter Arner and Mikkel Ryden find in their group? Well, when they split them into obese or non-obese, a dichotomy here, we see that the obese patients produced the bone marrow-derived adipocytes at a significantly higher amount than compared to the non-obese group. And when they broke it up by BMI into lean, overweight, obese, or severely obese patients, you can see the sample size goes down as it goes up, but you still see a really big stepwise increase that's statistically significant between these different groups, showing that obesity status, although not just having more of these cells because they maybe have more adipocytes, but as a percentage of their overall adipocyte population, this is still going up. So maybe some of the sequelae that's associated with the negative metabolic phenotype of obesity is due to more of these cells being present in these patients. And then looking at a correlation of donor cell production rate as a percentage on the y-axis and BMI on the x, you can see again a nice significant correlation. As BMI increases, so does the percent of cells that are coming from donor DNA. So this was really the first work in obesity. We haven't, our sample size was really small, and so we weren't able to break up anybody by BMI or obesity status. We didn't have a very big range, so we didn't see much to this effect in our group. One other really consistent observation that Dwight has made in his animal models is that he consistently sees that the female mice accumulate or produce more bone marrow-derived adipocytes than the male mice. So we all know the importance of making sure that we have equal representation of male and female animals in our studies now, depending on what we're looking at. And so by him doing this, he was really able to see this interesting divergence. Now, the males and females have similar distribution patterns. So still the highest in the gonadal region, followed by perirenal and subcutaneous. But the males consistently in the open bars here have a lower production of bone marrow-derived adipocytes as compared to the females. Although only reaching statistical significance in the gonadal region, you can see the tendency throughout the other two depots. So this was really interesting, and this was the set of data that my mentor, Wendy Court, the clinical researcher, saw and said, we need to study these in humans. We need to understand, do females have more of these than males? So we started a collaboration together. We did our first initial proof of concept study as the bone marrow transplantation. And then we took a step back, going back to the animal models to try to determine how do sex hormones impact the accumulation of these cells. So the next slide is just going to be female animals. So we recently finished a study, and we're just preparing these for publication, where we took a group of animals and we had one group that were overectomized. So we can see here it's just a little bit different image, but this is the same luciferase activity, an in vivo image. The darker colors, the red and the yellow, indicate an increased accumulation or production of the bone marrow-derived adipocytes. So we see a simple overectomy. This is, I think, 10 weeks after overectomy. You see a much greater production of bone marrow-derived adipocytes as compared to the wild type. We also used an ER-alpha knockout. This is a whole body ER-alpha knockout, and observed similarly when you take away the estrogen signaling through ER-alpha, you also see a similar increase in production of bone marrow-derived adipocytes as was observed in the overectomized animals. When we added estradiol back to the overectomized animals, we saw a reduction at the whole body level back to or similar to the wild type animal. So we really do think that ovarian hormone status, specifically estradiol signaling through ER-alpha, is playing a role in the production of these cells. So we have a little bit more to work out. I think some people might be thinking, well, your previous slide, you showed that females had more than males, and now you're showing you take their female hormones away and their production goes up. So we have a little bit more to study on that, and we really need to hone in on the male and understand how testosterone is impacting the accumulation of these cells and the testosterone conversion to estrogen. And so I'll show you in my future and kind of ongoing studies that I am interested in this and what models we're using to study that. But that is a little bit of a, we were a little bit surprised that overectomy increased it so much when we were seeing females seem to accumulate more of these. So we have a little bit to work out there still. So when we took the adipose tissue depots out of these animals, we did a luciferase enzyme activity assay at the depot level. And here you can see in the black bars is our wild type, the red bar is the overectomy animal, and the purple is the ER-alpha knockout. So a higher bar means more luciferase enzyme activity, which means higher production of bone marrow-derived adipocytes. Similar to what we saw in the whole body level, overectomy increased the production of BMP-derived adipocytes, similar to ER-alpha knockout, and that was both in the gonadal and subcutaneous region, although, again, the gonadal is really showing a much higher production of these cells. When we add back estradiol, it's brought back down to or below baseline level or wild-type levels, but adding back progesterone does not have the same effect. So we really do think there's an estradiol-specific effect going on here, estrogen signaling through ER-alpha impacting in some way, the development of these cells. So how do we continue these studies in humans beyond the bone marrow transplantation population? So it's great that we're able to bring in this clinical population, but there's a lot of limitations that go along with this. And we want to be able to bring in anyone who wants to volunteer for a study off from the street, take an adipose tissue biopsy, and understand what percentage of their cells is coming from what lineage. And if we can determine that, can we understand how the accumulation of these cells are associated with different disease risks? So the major limitation, I just want to remind you guys here, is that the surface markers that we know are usually expressed on bone marrow-derived cells no longer exist on these cells once they go through their transdifferentiation from a myeloid to a mesenchymal-like cell. So what are the strategies we've been working on to try to identify a biomarker, and how am I continuing to study these cells in humans even though we have some good limitations? Because my mentor, Dwight Clem, he's continuing to study these cells in animals, and if I want to pursue a career in this area, I need to find a way to study these in a clinical population where I can have a research program of my own to stand on. So I have developed a human primary cell culture model that I've been using right now to continue these studies in humans while we're working on identifying a biomarker. So what we do is we take an adipose tissue biopsy from a healthy human. We separate out the mature adipocytes from the stromal vascular fraction by a collagenase digestion, centrifugation, and these are the stromal vascular fraction is all those non-adipocyte cells that are in the adipose tissue. So we spin them out, we then analyze them by flow cytometry and cell sorting, and we're able to isolate out different populations of conventional versus myeloid lineage cells. So we do our cell sorting, our mesenchymal lineage cells, and I'll have a slide later that shows the surface markers we're using to identify what the mesenchymal lineage cells are. We can plate them on plastic, proliferate them, and grow them just like you read about in any study that's doing adipose-derived stem cell culture. On the other hand, when we isolate out our myeloid lineage cells, these cells, we can put them on plastic, but they don't grow the same way that a mesenchymal lineage cell grows. So if we give them the same growth factors that we would give our regular mesenchymal lineage cells that are isolated from the adipose tissue, those cells just kind of sit there and they don't grow or differentiate. So Dr. Clem's lab came up with a fibrin clot or a 3D culture system that allows us to suspend these cells in an environment that we think is more similar to what they would experience in the adipose tissue. So experience some tension and different things that would maybe push them down the line towards this unexpected differentiation pathway. So we take these cells out of the flow cytometer, we suspend them in fibrin for 5 to 6 days with growth media over the top. We can actually see them start to grow in there. About 6 days later, we digest them out of the fibrin, plate them on plastic, and we're able to see them become plastic adherent and grow. These are not going to show up well here, but proliferate like a usual mesenchymal stem cell would, differentiate into an adipocyte as measured by an oil-redo assay. So we can see that whether these cells are coming from a myeloid or a mesenchymal lineage, we're able to get them to behave like a mesenchymal-like cell in culture. So we've been able to now do this in mouse cells, and this is kind of the basis for my new KO1 award, where we're trying to find ways to study these cells and understand their clinical relevance beyond having a biomarker. So what am I doing with this culture system? Well, first thing in my primary really need is to determine a biomarker to identify these cells in all humans. So I'll talk in a minute about the preliminary data I have for that. We also want to determine the metabolic phenotype of these cells. So I've said we've done gene arrays, but we don't really know what that means as far as the metabolic phenotype of these cells. Just because they have low mitochondrial enzymes doesn't mean that their mitochondrial content or function is going to be lower than a regular adipocyte. So we really need to figure that out. And then I'm really interested in determining how age and or sex hormone status impacts, number one, the accumulation of these myeloid progenitors in the adipose tissue, as well as their propensity to go down this unexpected differentiation pathway. So whether women have intact ovaries and have normal menstrual cycles compared to the postmenopausal state, and then in men, if they're young and have high testosterone levels as opposed to older and may have slightly lower testosterone levels. So first, let me talk some preliminary results from how I'm using this to identify a biomarker. So to date, we've been using a flow cytometry strategy, and this is just a snapshot, and I'm not going through my entire flow strategy, but wanted to show you how we can use the CD45 and CD14 myeloid marker to get a really neat idea of what different cell populations might be in an adipose tissue digest. So again, this is the stromal vascular fraction. We're just looking at single live cells. There's a number of gating that we do before this step. And I'm able to isolate out conventional lineage cells. So these are mesenchymal lineage. These are CD45 negative. And then there's a variety of beliefs out in the literature on exactly what a mesenchymal lineage cell as far as a pre-adipocyte is from a human. But we think it's pretty well settled now. It's CD31 negative, so these are not endothelial cells. And 34 and 29 positive. So I sort out this population. There's a few more gates beyond this that I'm not showing you. We'll sort out that really specific conventional mesenchymal lineage cell. Then I also isolate out 3 of the many different myeloid or hematopoietic lineage cells that we can identify, and identify them by a number of different surface markers, and you can see they show up as different populations here on my flow. And we can talk more details about this if you want later. I just wanted to show you as kind of, I was brand new to flow cytometry, and I think this is like the neatest technique to be able to do to sort these cells. So I have a variety of other myeloid lineage cells, including macrophages, monocytes, and neutrophils from the adipose tissue that we sort, put through the fibrin, and then are able to differentiate into what look like fat cells. So what I've been using is a proteomics approach in order to analyze the cells that we're growing in culture. And so far, this is super preliminary data. It's just from samples. They're all from one patient. So this is an N of 1, but they're different myeloid accumulation populations that are all from the same patient or versus conventional. And what we found in our proteomics analysis is that there is evidence that all of the major adipocyte proteins we would expect to see in an actual adipocyte, and not just a myeloid cell taking up lipids, are present. So we see adiponectin, perilipin, LPL, HSL, CD36, a lot of these proteins that we know would definitely be expressed in a mature adipocyte. So I was very excited about this to show that the cells I'm differentiating in culture, specifically from the myeloid lineage, that would usually not express these markers, have these adipocyte-specific proteins. So I was also interested in seeing, okay, well, how many proteins do we detect in these? So we had 4 samples, so the conventional, the monocyte, macrophage, and neutrophil. So in all, we detected 2,474 proteins. So whether it's expressed in just one or all 4, that's the total number of proteins that we detected in these 4 samples. We found 376 of them were only expressed in the conventional mesenchymal lineage cells. So narrowing things down a little bit. These are markers that are only in conventional cells. And 135 were only in the myeloid cells. So these proteins were expressed in all 3, monocyte, macrophage, or neutrophil differentiated adipocytes. So we were really excited to find this many proteins to start honing in on and determining, are any of these potential candidates? So when you look at these 135 proteins, I found a publication that has identified the surfaceome of the adipocyte. So these are proteins that would be expressed on the protein of an adipocyte. And I'm really interested, although not only looking at a surface marker, I want to try to find a surface marker for these adipocytes so I can digest these tissues and use the flow cytometry strategy to really get an easy way to say 5% of these cells are from the bone marrow and 95% are from mesenchymal, and even maybe toss in an endothelial lineage marker. So I was able to compare the adipocyte surfaceome that was previously published by this group here at the bottom and find that 7 of the proteins out of my 135 are found on the surface of adipocytes. So this was really cool. Here's the list of the 7. They don't really mean much to me yet, but it at least gave me hope that this technique is going to work in identifying some proteins. So next, I need to repeat this in a number of other samples to show that these proteins are consistently showing up in these cells. This first sample was from a young premenopausal woman, and so I'm working on collecting samples from a postmenopausal woman, an older male, and a younger male to age match and kind of get a wider variety of populations so that we know that this isn't just a sex or age specific protein content, but that we are actually seeing that these could be potential candidates. So I'm always interested in feedback, or if you guys have experience in other techniques that you might think be able to help us identify a different biomarker for these cells, it would be greatly appreciated, but this is kind of the avenue that we're going down right now. So what about the metabolic phenotype? How are we pursuing that? And I'd really love to present some data on this very soon. So I'm conducting in vitro studies of these differentiated adipocytes, and I'm going to do our bone marrow compared to the conventional mesenchymal. So I want to show that we're showing the characteristics that we have been published previously in our conventional cells compared to the bone marrow derived. We're analyzing conditioned media from the cells, doing in vitro insulin sensitivity, such as lipolysis and glucose and free fatty acids, mitochondrial content and activity, as well as metabolomics analysis on these cells. And then we're also continuing the mouse studies to understand the metabolic phenotype in an in vivo situation. So Dr. Clem is developing ablation strategies where he can have mice that develop the bone marrow-derived cells as they normally would, and then other mice that will not develop the specific lineage of cells, and then determine, does their survival different, is one group healthier and live longer, and then we also want to do metabolic cage measurements, tracer studies to really try to understand what is the metabolic phenotype of these mice. And then to get down to the core of the matter of where I really want to see this research go in the future as far as my lab is concerned, is really understanding the influence of age and sex hormones on the production of these cells in humans. So I have a number of different studies that I'm obtaining adipose tissue biopsies from right now and generating number of cells to do these studies on. So I have a cross-sectional study of older and younger men and women. So the women, it's very easy, stratify pre versus postmenopausal. The men, we're kind of age matching with the women, and we are measuring sex hormone levels, so testosterone, estrogen in men, as well as the estrogen and progesterone in the women, and FSH and LH levels to categorize their menopausal status. So we're getting some cross-sectional samples there. I also am able to obtain adipose tissue biopsies from premenopausal women who are undergoing an GnRH agonist therapy. So this is gonadotropin-releasing hormone agonist therapy. So Dr. Court has a large intervention study ongoing, and a couple other people at our university use this intervention. What it does is it suppresses ovarian hormone levels so that it kind of gives women a preview of menopause. So it's used clinically for treatment of fibroids and in vitro fertilization, periods when you really want to exquisitely control hormone levels. But in this case, we're using it just as a clinical intervention to study a variety of body composition, physical activity, energy expenditure changes. And I'm lucky enough to be able to obtain adipose tissue biopsies from the belly and the thigh of some of these women in order to isolate out the different populations in the adipose tissue and grow these cells up in culture. I also am able to obtain adipose tissue samples from a similar intervention that's ongoing in men. So this is only a 1-month intervention. So these men are getting a GnRH antagonist therapy. So they're getting their hormones suppressed. And then some men are getting testosterone replaced. So they're getting their testosterone brought back up to normal. And other men are getting their testosterone replaced, but also getting an aromatase inhibitor. So this is to stop the conversion of testosterone to estradiol and try to tease out what's going on at the sex hormone level in men, because we always think testosterone is the male hormone and it controls everything, but we know that estrogen actually has an important role in men, especially in regulating adiposity. So 1 month is pretty short, and we're not sure what we're going to get yet, but we do know that within 1 month you already start to see changes in the immune cell accumulation in the adipose tissue. So we're going to sort out these cells and see what kind of changes that we can get after a month. The other part of these male and female interventions is that I have a series of experiments where I'm going to expose these cells to different amounts of sex steroids in culture to recapitulate the in vivo environment. So if they have low sex steroid environment in this person that I took the cells from, I'm going to expose them to low sex hormone, and vice versa. If it's from a premenopausal woman, I can take some of the cells and expose them to a certain amount of estradiol in culture. So these are all plans ongoing. I'm currently obtaining the adipose tissue samples, growing up the cells, and getting the training as part of my career development award to really learn how to do all of these in vitro studies. So my overarching hypothesis in this area is that we think the loss of estradiol leads to the accumulation of bone marrow-derived adipocytes, and that is associated with an increase in visceral adiposity. So we know that the loss of estradiol increases visceral adiposity. So we know that this link is there. And what we're hypothesizing is that this step happens in the middle. And one of the reasons that we see specific increase in visceral adiposity is that it's the increase in production of these cells with the menopausal transition that's occurring. This also lines up with the gene expression fingerprint that we see in these cells thus far. So low leptin levels, we know increased visceral adiposity is associated with lower leptin. We also know that visceral adipocytes tend to release more inflammatory cytokines. And then mitochondrial enzymes, we're not so sure, but we think it fits into the reduced energy expenditure that we've observed in some of our clinical studies of women with the ovarian hormone suppression. So we think that these really align well with this kind of overarching hypothesis. So we have a lot of work to do to make sure that this is really what's going on, but we think it's very promising. And then we all know that increasing visceral adiposity increases cardiometabolic disease risk. And so this is how we think shifts in the cell population of adipose tissue actually might be impacting overall disease risk. So you might have some adipocytes in the tissue that have some characteristics that are coming from mesenchymal lineage, and some adipocytes in that tissue that have the characteristics coming from a bone marrow derived origin. So to summarize, we know that adipose tissue is critical in maintaining metabolic homeostasis. We also now know that multiple progenitor sources from adipocytes appear to exist. So we know mesenchymal lineage is definitely a development pathway. We also now believe that endothelial cells might be contributing to this. And now our research suggests that myeloid lineages also can contribute to adipogenesis. We also think that these cells likely contribute to the functional diversity of distinct adipose depots. So why fat from the hips and thighs is different from that in the subcutaneous belly region versus the visceral belly region. And then I think that the adipocyte composition of adipose may be a mechanism by which this regional adiposity is impacting disease risk. So where does this lead me in the future? Well, biomarker identification, I've emphasized that a few times. We need to identify one, and hopefully we're close to that. We also know that obesity and the loss of ovarian hormones are two factors that influence the production of these cells, and they both increase it. And then I'm interested in knowing if accelerated production of these cells is a mechanism under which central fat depots increase with the menopausal transition. And then I really think that understanding adipocyte lineage, whether it be cells of the mesenchymal, myeloid, endothelial, brown, beige, there's all these different types of cells. If we can figure out exactly how these cells develop and what are the factors that influence their development, I think we can come up with better strategies to manage obesity-related diseases, and may give us some underlying clues as to differences in people with healthy versus unhealthy obesity, or lean people who have extreme metabolic disease for some reason. Maybe if we're able to analyze their adipose tissue, we can see that they actually have a very different cellular composition than a lean person who has a healthy metabolic profile. So I ultimately would like to start to be able to develop interventions that are aimed at promoting or attenuating the development of different types of fat cells. And so, as said at the beginning, my background is exercise physiology, and as you can tell, none of this research has anything to do with exercise. So I'm really interested in exercising some of the mice and getting some adipose tissue samples from humans who are either well-trained or not trained or undergoing an exercise intervention to understand if women stay active through menopause, is this the way that we can kind of control the different types of fat cells that are developing and thereby hopefully reduce their metabolic disease risk. So just some acknowledgments. Again, I'm in the Division of Geriatric Medicine in the School of Medicine at the University of Colorado. My mentors here, primary mentors, are in red. So Dr. Kort's in my division and Dr. Clem is in a different division. And I have also a number of other faculty members on my mentoring committee for my KO1. And so a lot of people help me out with this, and especially the staff at the Flow Core and in Dwight's technicians. So I think that's it. So any questions you guys have, I'd be happy to take.
So you got a really good dichotomy here. So there's a recent study that came out that women who've been diagnosed with estrogen receptor hormonally responsive breast cancer, they are, initially the idea was to stay on aromatase or tamoxifen for 5 years. Now it's pretty much you can be on for the rest of your life. So that's going to, so you have women that are on this for 10 to 20 years.
Yeah.
So you're dropping estrogen.
Yeah.
Completely. But then you're putting into, of course, there's other work, our colleague here, that's saying, well, now you got an inflammatory environment.
Yeah.
So what do you think, what's balancing out here? Is there any evidence of women that are on these aromatase inhibitors, do you see a change in, or is there other diseases that are associated with tamoxifen or aromatase?
Yeah, that's a good question. And I remember when I